Lifestyle factors such as smoking and diet, along with genetic background, can significantly increase the risk of cardiovascular disease. Cardiovascular disease is caused by the formation of plaque in the blood vessel walls. This is known as atherosclerosis. This disease silently progresses by thickening the blood vessel walls, leading to a reduction in vessel diameter and the formation of blood clots. These blood clots can obstruct blood circulation and can trigger a heart attack or a stroke. A monocyte is a type of white blood cell, which forms part of the body's defense system. Monocytes circulate freely in the bloodstream. To fight infections or repair damaged cells, monocytes migrate from the bloodstream into tissues. To do so, monocytes bind to endothelial cells that line the interior wall of blood vessels and migrate through small gaps. In homeostasis, the influx and efflux of monocytes between the blood circulation and tissues are balanced. Smoke exposure has been shown to promote endothelial dysfunction and stimulate inflammatory processes. These, in turn, enhance the influx of monocytes from the blood circulation towards the tissue. These monocytes in the vessel wall may differentiate into macrophages that take up a large amount of lipids and transform into foam cells or die. Both situations lead to a reduced efflux of these cells back into the blood circulation, which contributes to atherosclerotic plaque formation. Philip Morris International is developing a range of potentially reduced risk products such as THS 2.2 that offer adult smokers a potentially less harmful alternative to cigarettes. Tobacco in a lit cigarette reaches temperatures of up to 900 degrees Celsius and produces thousands of chemicals that are released and inhaled as smoke. Heating tobacco at approximately 300 degrees Celsius produces an aerosol that contains a significantly lower number and level of harmful constituents. It is important to conduct extensive and rigorous scientific studies comparing the biological impact of THS 2.2 with that of a reference cigarette on a mechanism-by-mechanism basis. For example, in vitro laboratory models enable the measurement of several processes affected during plaque formation, such as endothelial cell barrier function, transendothelial migration, cell death and inflammation. A specific in vitro study was designed to assess the impact of THS 2.2 on these processes compared with that of a reference cigarette. First, smoke from a reference cigarette and aerosol from THS 2.2 were generated using smoking machines. The smoke and aerosol were then dissolved in an aqueous solution through bubbling, and water-soluble chemicals in the aerosol were captured in the solution. Different concentrations of this solution were then used to treat cells prior to the 4 assays. Our results show that cigarette smoke damages the barrier formed by endothelial cells, which would increase the risk of atherosclerotic plaque development. It was found that the aqueous solution of the THS 2.2 aerosol has a lesser effect on barrier function. Its concentration would need to be increased 16 times to observe effects similar to the ones induced by the aqueous solution of the reference cigarette smoke. Transendothelial migration or efflux was then assessed. Decreased efflux would indicate an increased risk of atherosclerotic plaque development. The results showed that the aqueous solution of the THS 2.2 aerosol has a lesser effect by 18 times on monocyte efflux than the aqueous solution of the reference cigarette smoke. The effects on cell death were then assessed. Increased cell death also leads to an increased risk of plaque formation. Results show that the aqueous solution of the THS 2.2 aerosol is 26 times less potent at inducing cell death than the aqueous solution of the reference cigarette smoke. Finally, the effects on inflammation, which is a marker of plaque formation, were assessed. Results show that the aqueous solution of the THS 2.2 aerosol is 10 times less potent at inducing a release of monocyte-derived molecules causing inflammation than the aqueous solution of the reference cigarette smoke. In summary, this study has shown the potential for THS 2.2 to reduce effects on monocytes and endothelial cells in processes relevant to atherosclerosis compared to the reference cigarette at matching nicotine concentration. This data indicates that THS 2.2, a heated tobacco product, has the potential to positively influence factors known to be associated with cardiovascular disease. Other scientific studies are ongoing or have been completed, and before making any claims about reduced risk products to users, we will rigorously evaluate the full set of data generated from these other scientific studies. Please read our full publication online to find out more about this study.